Friends, welcome to Biology Made Easy. Today we will discuss some of the Mendelian disorders. After Mendel, it was found that due to defects in the gene, several disorders, several diseases are occurring in different organisms, including the human beings. So these genetic disorders are mainly of two types. They may be Mendelian disorders or they may be chromosomal disorders. The Mendelian disorders, they usually occur due to mutation, which may be deletion or addition of some of the important genes. While the chromosomal disorders, they occur due to change in the structure of the chromosome. That means the genes are reshuffled, the nucleotides are reshuffled, leading to deletion, addition, insertion, and translocation. So these Mendelian disorders, they inherit according to Mendel's laws, but their expression sometimes may be known phenotypically, may not be known, but we can trace where that particular defect lies by analyzing the pedigree of a particular family. The different Mendelian disorders, they include sickle cell anemia, hemophilia, color blindness, thalassemia, alkaptonuria, phenylketonuria, albinism. All these are coming under the Mendelian disorders, and we will have discussion one after another. Today in this video, we will discuss the sickle cell anemia. Sickle cell anemia. Now what happened? In 1904, one doctor known as Dr. James Eric, he was examining a patient who was 30 years old and was black. And he observed that the patient is suffering from a lot of symptoms like a fever, headache, dizziness, enlarged heart, then damaged kidney. So many symptoms are there. And when he examined the blood of that patient, then he was surprised that some of the RBCs of that patient were sickle-celled. Because usually the normal hemoglobin is round and biconcave, but in that patient he found some of the RBCs are elongated and sickle-celled. Again, the hemoglobin content was only 50%. That means the patient was also suffering from anemia. So in this way, the RBC was sickle-celled and the person is suffering from anemic condition. So in this way, the sickle cell anemia was discovered by Dr. James Eric. Then in 1949, another scientist, Linus Pauling, he was studying the structure of hemoglobin, because the normal hemoglobin usually the normal hemoglobin, which is responsible for carrying oxygen in our body, it is this round and biconcave shaped. But he found that in case of all adults, it is normal and is designated as HbA. But in case of sickle cell anemia, it is designated as HbS. Now what is the reason behind it? He found that the hemoglobin consists of 4 chains, two alpha chains or polypeptides, and each one consists of 141 amino acids. That means each alpha chain has 140 amino acids. There are also 2 beta chains or 2 beta polypeptides and each one contains 146 amino acids. So these 4 chains to alpha and to beta, they constitute the hemoglobin. But what happens? There is a little bit change. In case of the normal hemoglobin, the structure of the DNA at one point, that is GAG and CTC, and in this case it is GTG and it is CSC. That means this is the structure of the DNA. Now in this case what happened? This thymine is replaced by adenine. So that this is CTC, the corresponding is GAG, but since it is replaced by A, so naturally it is GTG. Now when it will give rise to mRNA, the mRNA, here the mRNA will be GAG and here it will be GUG. Now according to genetic code, this GAG will give rise to glutamic acid, while this will give rise to valine according to genetic code. This glutamic acid is usually present in the 6th position of one of the beta chains, while the other ones, this is the amino acid number 5, 4, 3, 2, like 1. So 1, 2, 3, 4, 5, 6. At the 6th position, there is glutamic acid there. When this is replaced by, again this is the 6th position, and in this side also we will get amino acid 5, 4, like that amino acid 1, and this side we will get amino acid number 7. That means at the 6th position of one of the beta globin chains, when glutamic acid is replaced by valine, the normal hemoglobin is converted to sickle cell hemoglobin. So this is a case of substitution mutation. It is a case of substitution mutation. It is also, now the thymine is replaced by adenine. Thymine is a pyrimidine and this is adenine. So it is also a transverse mutation. So this is the basic reason, or this is, you can call this is the genetics of sickle cell anemia. Now, if a person is suffering from sickle cell anemia, then what will be the different symptoms? Now usually among the symptoms you will find the RBC. RBC is usually sickle shaped. This is number 1. The second point is that there is a 50% of hemoglobin. That means the person is suffering from anemic condition. Now, the next thing is, what will happen? When there is a low oxygen concentration, then there is a problem. Because 50% of normal hemoglobin is there and the 50% the sickle cell is there. So when there is a low concentration of oxygen, as in case of high altitudes, there will be pain. So this pain may be in legs, in joints, or in abdomens. And sometimes it may be very severe also, but these symptoms are found only in persons who are suffering from sickle cell anemia, and that sickle cell anemia is found only in case of homozygous condition like this. Because the sickle cell anemia is an autosomal recessive disorder. So only when it is present in homozygous condition, the person will suffer from it. Now, how we can avoid all these things? And again, another thing, how this sickle cell anemia persons, how they come to the society, because all persons are not the not suffering from sickle cell anemia. So what is happening, let us discuss. If two carriers, one father and one mother, there will be marriage between them, then what is happening, you see. Suppose the father is heterozygous, HBA and HBS. The father is heterozygous, and also the mother is heterozygous. Now the father will produce two types of sperms. This is the normal one, and this is the sickle cell type. The mother also will produce two types of ova. This is the normal type, and this is the sickle cell type. Now, when this will fuse with this one, then it will be HBA and HBA. That means this person is completely normal, like all of us. But when this will combine with this one, then it will be HBA, HBS. And when this will combine with this, again it will be HBA and HBS. That means these two are sickle cell carriers. And when this will combine with this one, it will be homozygous, HBS and HBS. This is the homozygous. That means this is normal, these are carriers, and this is the person who will suffer. That means this person is sufferer. That means 25% is normal, 50% are carriers, and 25% is sufferer. That means 1 out of 4 children have a possibility of becoming the sufferers of sickle cell anemia. So, how to avoid it? That is again important. We can avoid it by examining the blood samples before any marriage proposal. Suppose there is a marriage proposal between the two persons, one girl and one boy. If both of them are heterozygous, in heterozygous condition, that means they are carriers. So naturally, 25% of their children will suffer from sickle cell anemia. So if that can be the marriage between the carriers can be avoided, then the sufferers can be avoided. Then you may ask one question: how we can know that they are carriers? The answer is very simple: by examining their blood. If the blood will be examined, then we will find two types of the hemoglobin in them. That means 50% are normal and 50% sickle cell. In that case, it will be advised, don't marry, because your children, there is a possibility that your children may suffer from this. So in this way it can be avoided. Then another aspect of this sickle cell anemia is malaria. Because there are certain areas like Africa, Mediterranean areas, and even in some parts of India where people suffer from malaria. So in such cases what happens, usually those who suffer, those who are carriers of this sickle cell anemia, they can tolerate it, while the homozygous conditions, they are unable to tolerate it. Let us discuss how it is happening. Now suppose in case of sickle cell anemia, suppose one is homozygous HBS. So in this case what will happen? The person will die due to sickle cell anemia, because sickle cell people, they die when they be adult. Before they attain the age of 20 years, usually most of them die. So when it is homozygous condition, that means the person is suffering from sickle cell anemia, and there is every possibility that will die. Suppose it is homozygous normal. When it is homozygous normal, there is also every possibility that the person will die due to malaria. Because in malaria prone areas like Africa, Mediterranean and some parts of India, those who are homozygous normal, in them this malaria parasite plasmodium, it can multiply and it may cause death. But if a person is heterozygous for these two genes, heterozygous, then the person is carrier. That means these carriers, they will not die due to sickle cell anemia and they will not also die due to malaria, because the malaria parasites cannot complete their life cycle within these carriers because of the sickle cell RBC. So this is known as selective advantage. It is known as selective advantage, and it is also, you can call it as heterozygous superiority. Heterozygous superiority. That means those who are heterozygous or those who are carriers for the sickle cell anemia, they have selective advantage. They are superior. They can survive, they can reproduce, and they will not suffer neither from the sickle cell anemia nor from the malaria. So this is one of the advantages with the carriers. So this is all about the sickle cell anemia. Now let us discuss questions. So MCQ 1, in sickle cell anemia, one choice, glutamic acid is changed to valine in alpha chain. Second choice, valine is changed to glutamic acid in alpha chain. Third choice, glutamic acid is changed to valine in beta chain. Fourth, next choice, valine is changed to glutamic acid in beta chain. This is one question. Now let us discuss the second question. The second question is, sickle cell anemia is a case of first choice deletion, second choice insertion, third choice transition, and fourth choice transversion. You will be able to answer both the questions because we have discussed all those things. So thank you very much for watching it. God bless you. Have a good day.